to start out talking about sunlight. Let's talk about UV and sunlight and how UV is in natural sunlight and how different things are treated with UV light. Some of the things that are interesting is how not enough sunlight because of the UV will lead to rickets. And another thing that's being UV is used in hospitals to help babies with jaundice. Is that correct?
Well, absolutely. Well, of course, it's used in food production, hospital sterilization, food sterilization, sewage treatment, water purification. In our case, blood treatments for deactivating and killing virus.
Yep.
And blood sugar.
And not only viruses and bacteria, but it's also one of the only known ways to inactivate snake venom. Is that correct?
Absolutely. Snake venom, we're using the ultraviolet primarily now for treatment in the veterinary industry for infections, cancers, all kinds of things, because we don't have the restrictions in the veterinary industry that we do with the FDA, for example, for treating people, but it's shown remarkable effectiveness, particularly in even viral and bacterial diseases. Animals get them too.
Yep, sure. Now, why is blood irradiation not in the mainstream at this point?
I think a couple things happened. Back in the 1920s, when Emmett invented the non-hemo irradiator, he called it ultraviolet blood irradiation. Irradiation is not a friendly term. I think he kind of screwed up by calling it that in the first place. But his primary focus was helping women who got septic infections from abortions back then. They were quite crude. And also to fight polio and other diseases at the time. They didn't have AIDS. They didn't have viruses that are ravaging the world today. They didn't have all these variants that we do have and all these mutations and mutagens. And if we would have had it or used it for example, the Spanish flu, or they would have had it back in the Middle Ages when they had the Black Plague, it wouldn't have been an issue. But what happened was the medical industry came up with antibiotics. Sometime in the 40s, and the press just piled right on and said, oh, we found the answer for disease. We've got a cure for everything. And of course, the treatment that takes an hour or so, and you treat blood and take it out and put it back in the body became pretty much relegated to the back room.
Yeah, people could just pop a pill and boom, they felt like a few pills.
Healing in a bottle is great.
Yeah, it makes it easy. Yeah, so you're talking about history a little bit. You want to go more into, you mentioned somebody a minute ago, and the history of where this started and what their successes were. I guess they treated thousands of people back
There
in
were
the
thousands of people treated. Hannerman Hospital in Philadelphia was one of them. Emmett Knott was in Seattle. They treated things there. Robert Olney, Robert Hancock, Dr. Miley. There were so many people, and if you go to the Library of Congress, you can look up these treatment cases and clinical results. They're all in the Library of Congress. It shows that this has been in use and effectively treated diseases, multiple diseases, decades and decades ago.
Yeah, that's amazing.
That was before the institution of the FDA.
So some of the things that they were treating back then were like, sepsis and toxemia?
Septicemia kills 80,000 people a year in hospitals. Unfortunately, we're not able to treat people in hospitals with septicemia, but my feeling is that 2 35-minute treatments with patients day 1, day 2 would resolve most of these septicemia cases.
Wow,
I'm not trying to start an argument here, but we're just saying that historically this has shown to be effective.
Yeah, that is amazing. Now, some of the mechanism behind this, I've been doing some reading, and I wrote a book about light medicine, and some of the stuff that I've found is that it raises the oxygen levels there's a detoxifying effect, but then what it does is it raises the blood oxygen level as much as 50% an hour. Is that accurate?
It can do that, but the primary action of ultraviolet is to damage the DNA of virus and bacteria in such a way that they're unable to replicate. To get slightly scientific, you know, it causes covalent bonds to be formed in adjacent pairs of DNA of virus and bacteria, making them unable to replicate. It inactivates them or kills them depending on the amount of joules, which is watts per second, you know, that are delivered to the pathogen. The beautiful thing about it is the way God designed pathogens is He gave them a thin outer shell which enables the ultraviolet to penetrate that easily, while at the same time we have a protective mechanism in our body's own cells that cause the pathogen to not, or the ultraviolet to not affect.
It's just basically supporting our cells. It's not any
supporting the immune system.
Supporting the immune system.
It's an
supporting
alerting the
cells.
immune system too, because once you unmask those invaders and you let the immune system see them, then a healthy immune system, otherwise healthy immune system, can identify them and attack.
Now, does it kill these pathogens activating? I read something about activating antigens. Then the body sees these antigens from some of these dead toxins, and the body actually, it's like an inoculation effect where the body sees it and is able to attack the rest of the cells. Now, how much of the blood is actually pulled out of somebody when they're being treated?
Less than 5%.
And not much.
You know, it's not a lot, but what happens is you have this autogenous vaccine effect. In other words, when you give someone a vaccine, you're giving them, basically you're giving them dead pathogens in some mixture of substances. So that's designed to alert the immune system to their presence. And if the body sees any of them, the immune system sees any other ones that come in, they say, oh, wait a minute, that's a bad guy, let's go get him.
Okay, and another is, so what diseases have you found are most susceptible to this ultraviolet blood irradiation? Is there, I mean, because we know that the virus is out right now, and it's only a single-strand RNA. And I read an article that said that single-strand RNA don't stand a chance against UV light. Are there other?
Well, but not only that, but they have a lipid membrane. They have a phospholipid membrane, and viruses with that type of structure are easily rendered inactivated by this. For example, we did some treatments in Dominican Republic for Zika. Zika was around in Florida 3 or 4 years ago and scared everybody, and it was a lot of hysteria about it for a short period of time, but when we took the hemolumin over to the Dominican Republic, we resolved, I guess it was 8 or 10 out of 10 cases in 2 days.
Wow.
We did a treatment day 1, day 2, day 3, they were well. And they looked at it as a miracle. Zika can resolve itself. The certain viruses that are out today, actually the coronavirus can resolve itself in patients that have healthy immune systems, but it has other components, the SARS component that cause respiratory distress and that sort of thing. But
well, and it's interesting because even when I wrote about light and published a book in 2012, I talk about how in a pandemic, this might be our only go-to situation because there's an antibiotic resistance now that's been building, and so I saw the ultraviolet blood therapy as a way to really help in a pandemic situation.
do you want to talk about the coronavirus?
We don't have to go directly. I mean, we can be more.
We can just talk about viruses in general.
Yeah.
The beautiful thing about it is, like I said before, the coronavirus, Zika, for example, the one that we were 100% effective with, is a lipid membrane virus. So is a coronavirus. There are a lot of viruses with that type of structure, and historically with those other viruses, we'd had virtually 100% cure success rate with, and so we think that we would have great success doing the same thing.
Well, I just interviewed
with
a
the
friend.
current situation.
I just interviewed a friend of mine a few days ago, which is up on the site, where he came back from Asia and he used your equipment, the hemolumin polychromatic device that you developed, and within 24, 36 hours, I mean, he was fine.
Right.
So it's pretty amazing what this technology can do. Now, this technology, like you said, has been around. There's been a couple people, like Dr. William Campbell Douglas wrote a book in 1993 called Into the Light, which the whole book basically talks about the technology that you've developed over here. And so it's a read that I suggest anybody to get. Now, there's another guy named Kenneth Dillon who wrote Close to Nature, and he talks about how chemiluminescence is, that our immune system is a chemiluminescent system.
Right.
And so all my research into the biophotons has kind of pointed in that same direction.
Well, we have photoreceptors in every cell of our body, so why did God put them there? God put them there so that wavelengths could stimulate biological process.
Yeah, and at the most basic level, as we know, vitamin D is a process through those same receptors.
Right, you go outside and get some vitamin D in a hurry. And of course, that's one of the great engines of the immune system.
But to clarify, those photoreceptors are in every cell in our body, and I just want people to
So
understand.
why do we have those there? Why did God put them there if they didn't serve a purpose?
Well,
and they do serve a purpose. And what we did with the hemolumin, if I can segue to that for just a minute here, is we call this polychromatic therapy because it not only has primary wavelengths that resolve the viral infection, but it promotes circulation by increasing capillary flow. Because one of the things with the virus that's ravaging the world today is it's a cardiovascular, it's a vasoconstrictor. So when you're constricting something, making it smaller, it makes it harder for blood flow and oxygen to get to the extremities of the body. So you have something that's a vasodilator. At the same time, we're producing nitric oxide. At the same time, we're protecting the SOD, the catalase of primary nucleotides in the blood, against any damage from ultraviolet insult, so to speak. And then we have the blue in there, which helps bacterial and viral inactivation. But the three primary wavelengths of ultraviolet we use propel what's called the Krebs cycle. And the Krebs cycle is NAD/NADH conversion, which is a cyclical energy that propels mitochondrial energy. And healthy cells are a properly charged cell is a healthy cell.
Nice. Yeah, that's important. So there's a system called photophoresis, which is approved by the FDA. It's a little different system than this, but photophoresis, if you want to talk about that, and it was approved, I guess, in the 80s or 90s. Talk about photophoresis.
Well, who's are you talking about? I mean, photodynamic therapy is what we're talking about, or photophoresis by taking the blood and treating it with light, returning it to the body?
Yes, that kind of photophoresis.
Well, they're using that at, Dr. Edelson at Yale University has developed a system that they call the, oh my goodness, it was actually by Johnson & Johnson, they call it the, slips in my mind, it'll come back to me soon, I'm old, that's what happens. The Theracos machine.
Theracos, yes, that's right.
And so they've treated, it's a little different in that they separate the white cells and the red cells in the body. They irradiate the white cells with a tremendous amount of energy, return them to the body, and they're doing this very effectively for a number of cancers.
So, and this has been out there for cancers, and
they're hyperactivating the immune system.
Yep, hyperactivating the immune system using light, fully approved by the FDA several years ago. Now,
decades ago.
Decades, yes, decades ago. So one of the things that's interesting is that, the different types of ways to do blood, UV blood therapy, there's several ways. And me looking into it and have written a book and articles, I've come across a lot of different ways. There's transdermal, which just UV waves are so short, I don't know if that would be effective. Then there's sublingual. What was his name? Tommy Perez at Harris Medical had it sublingual. What do you think about those two systems of treatment?
Well, you don't want to put a lot of ultraviolet light on the skin because you can cause skin cancer, so that's a non-issue.
So both
and it doesn't go very deep. Generally, wavelengths that penetrate in the body, have a particular resonance or, a way to wiggle through the tissue to get into the body.
Mm-hmm.
I mean, it's a dynamic, it's a mechanical thing.
So those two systems aren't that effective. Now,
well, Tom Perez's system was a good little system, I think. He had a ultraviolet sublingual system, and the nice thing about that is in the buccal region of the mouth, you have a tremendous amount of capillaries enough really to cover a football field. And you have maybe 1 micron to get to the blood, so when that ultraviolet can get there into the bloodstream, so you can radiate the blood through that tissue. But you can also cause burning of tissue. You can do certain other things. We prefer to use certain wavelengths of red light with photodynamic substances, molecules that can be activated, which are much gentler in terms of affecting
But I want to talk
skin.
about these different types of treatment. So we're talking about transdermal, putting it on skin. Then we're talking about sublingual. Then there's another guy, Weber, Dr. Weber out of Germany, that actually uses a fiber optic cable. Now I've heard pros and cons about Dr. Weber and this system. What are your thoughts about that type of blood radiation?
Well,
This is not using a needle, just for the viewer.
Too many
using a
needles
needle
with too many different
with a
wavelengths.
fiber optic cable that puts it right in the system.
right, but there's not enough energy in the system. That's the problem.
Okay. So that not enough blood is getting not enough energy to make it effective.
Viral inactivation is a function of energy over time and wavelength. If you don't have enough energy, you're not going to do anything.
Okay. So then there's other types of systems which uses a standardized cuvette, like I've seen out in the industry. So generally, some of the early devices were
Low-powered bulbs with a single tube.
yeah, with a single tube. So tell me about what your thoughts are on the single tube with the energy.
Once again, so if you have a 6-inch tube like many people use and a very low-powered bulb, let's say you've got an 8-watt bulb. Well, an 8-watt bulb will produce approximately 1 watt of ultraviolet. It
Okay.
consumes 8 watts of energy,
Yeah.
but it puts out 1 watt of ultraviolet. That 1 watt of ultraviolet is spread along this entire 6-inch tube, and it really doesn't get
So it's not enough.
illuminated from all sides.
Okay.
And even if it has multiple tubes, there's a lot of refraction that occurs, etc.
Okay.
So you don't get
Okay.
enough energy over time, because by the time the blood gets through that, it hasn't had time.
Now, what
to be
percentage of the
exposed.
devices in the marketplace are a single tube system,
All of them, except for ours.
except for ours? Why don't you go ahead and hand me that cuvette, if I can open that up, we can see
Sterilized
that.
cuvette, we use that in the
So this is a
veterinary.
sterilized cuvette. So the difference between the system, is it all right if I open this?
Yeah.
So the difference between this cuvette and other cuvettes in the marketplace is this has got 3 glass tubes that the
They're
blood, quartz tubes,
Fuse quartz
that
crystal,
the blood runs through 3 times in
So
front of the
it's-
light.
30 inches of contact.
Yeah.
So what we got
much
is,
more effective system than
well, the
what's in the
closest
marketplace.
system has a single tube.
Mhm.
It's
Yeah,
8
so most
millimeters
systems in the
instead of 6.
marketplace are just 1 tube,
Right.
and the machine that Gene has developed here has 3 tubes.
Well, the reason for that is obviously we want more contact time, but we also hit these different tubes with the different wavelengths,
Different wavelengths
of,
of
of the
Okay.
polychromatic visible
Mhm.
spectrum, as well as all 3 of them are being hit with our ultraviolet spectrum at all times.
Okay. Now, in debate with you on, I know there's flat cuvettes out there that are flat, and their argument is that a flat cuvette absorbs more light. What is your thoughts on this?
Well, that's okay. I mean, they're talking about direct radiation,
Mhm.
but the way we're set up is we have 4 legs of 2 long U-bulbs with ultraviolet, so we're getting 360-degree exposure on the tube, whereas they're only getting it from 180 degrees with a flat cuvette. They're not getting 360 degrees because we have 4 light sources. If you look at the cuvette as the center of an X, between those 2, that center cuvette and the 2 top legs, they're both being hit directly with ultraviolet light,
Okay.
as opposed to the middle one is getting hit directly. So we have a 360-degree illumination times, 3, so we have exponentially more light that's targeted on the cuvette.
And what kind of system did Knott and Miley, the guys back in the 30s and 40s that were doing this, what kind of system did they have?
It was a good system. They used a halogen bulb, which we analyzed the output of and approximated some of the wavelengths in there that are good. They also had some wavelengths that weren't good, because some people in current machines use UVB, which we don't use. We specifically don't use UVB because it can promote the formation and release of ANA's anti-nuclear antibodies, which could aggravate a condition such as lupus or fibromyalgia.
Okay, so what wavelengths of UV are you using in your
We're
device?
using 2 wavelengths of UVA and 1 wavelength of UVC.
Okay, so UVB has been proven to be bad, but aren't some of these other devices out there using? Is Theraclose using?
No, they use UVA only,
Okay, they only use UVA.
which is what people target for cancer therapies.
Okay, I got you. Okay, interesting. And so let's talk about the dynamic of using different compounds, like the photophoresis. Don't they mix that with metasoline, mop
Yeah, they use
So this
I
is
think
the one
they
that's
use
approved.
Mopate or Sorlins, et cetera,
So-
which are photoactivated substances which basically come from plants. You know, plants do photosynthesis, and they're very good photosynthesizers. You know, you've got chlorin C6, chlorin E6, you know, you've got epigallocattic and gallate.
Okay, so
epigallocattic.
what happens here is the light has some kind of a synergistic effect with these compounds?
It activates them.
It
It makes
activates
them more potent, and they become oxygen producers or pathogen, you know, destructive tools, et cetera. But they-
Because that's a specifically
hyperactivate their
light
activity.
sensitive compound. And
Right.
you've talked about using something called methylene blue in some of your systems or treatment.
Right, methylene blue is one of the first drugs in the U.S. pharmacopoeia. It's a blue dye, and it's it was discovered, I think, in 1886, and it's been used for a number of things. They use it for babies, you know, when they have oxygen deprivation.
Mhm.
They use it to resolve methemoglobinemia when people have found up oxygen.
So you're
in their body.
using this in conjunction with some of these light treatments?
Right, we use wrist pads, which we use with the machine, the hemolumin.
Why don't you turn on the machine? Let's see these
It's a
wrist
little
pads.
noisy, but we'll turn it
Okay.
on here.
Mhm.
There you go, it's plugged in, it's going through its usual thing. I'll just lift this up here, and we have some red wavelengths that we're using. We have 20 watts of red, which we target on the various legs of the cuvette. We have a green, which opens up the capillaries and promotes, I'm going to mispronounce it, the erythritosity of the blood. It helps
The viscosity of the blood.
it helps promote better blood flow.
Okay.
We have the 590 amber, which has been shown to increase nitric oxide promotion and release. We use a blue, which also has antibacterial capabilities to it that helps in balancing redux. We have 2 wavelengths of UVA in here, which promote which are one leg of the Krebs cycle, which is also good for antiviral, antibacterial. And we have your basic UVC, which is your
So those are the two,
germicidal lamp
UVA and UVC.
that people use for, you know, treating water and treating sewage and, you know,
Air
killing
purification.
air purification, that
Yeah.
sort of thing.
Yeah.
That's your most prominent one. Now, this is filtered out primarily in our atmosphere. A lot of the UVA is filtered out in the atmosphere. Some UVB gets through, and that's what's causing skin cancer.
Yeah, okay.
We don't use UVB at all
Okay.
because it said it
That
doesn't
makes
promote
sense. So
ANAs.
let me see the wrist pads over here. So these wrist pads, let me see this. So these are red light. So
Well, let me show you something interesting
that's a
about
lot.
this.
Look at that. That's a lot of light coming through.
Let me show you something interesting about this. What most people do, I don't know which leg this is, let me see. That's yours, okay, this is mine here. I'm gonna increase the power a little bit.
Okay.
What we do with it is we're simply trying to get to the blood flow in the wrist, the radius and the ulna. We put a photodynamic substance in the animal, for example, or in people in foreign countries. We're doing some research overseas. And what happens is that circulates in the bloodstream. So as it circulates in the bloodstream, it's going to pass through the radius and the ulna in the wrist.
Okay.
And what it does is every time it goes there, every time it goes through there, it's activated, so it becomes more active. But I want to show the camera something here. See, you can see with the power that we have, we can penetrate through the hand. we're simply trying to get into the bloodstream.
Now,
And I'll bet you, as my mom used to say, $1 to a donut when we get there.
let me see that for a minute. Now, I've been told that the light can actually affect the brain, people that have Alzheimer's, Parkinson's, traumatic brain injury. I mean, will this make me smarter, Gene? Can I actually increase
I don't think
blood flow?
anything can make you smarter.
Oh, thank
no matter how you try.
I'll take that as a compliment. I won't let him finish his sentence.
You know, that's right, you may be at the top of your game, you know, I mean, there's nothing that can make you better. But this is really good because what happens is when you photoactivate some substances, you actually add electrons to their outer orbital shell and they become more active, they become electron donors.
That's
So
amazing.
every time this passes through there, it picks up electrons and it donates them to viruses and bacteria and kills it.
That's amazing. But it does, there is a lot of evidence that it does support brain function.
Huh?
It supports... Let's give Gene a few slides.
All right, let's unplug these suckers, they're a little
well,
too strong.
I
But
hope
these are
people-
really critical in the aspect.
I hope people enjoyed watching this, because I think this is very interesting. Gene is one of the most interesting guys I know. Gene, you want to mention your website or any of this stuff?
ALDmed.com. You won't find a lot on there, but if you contact us, we'll give you some more information.
And
We
there-
don't like to be too wordy about it. But let me just say one thing here, if I
Okay.
can. We think that we have a really good approach to, let's say, another approach to the treatment of modern pathogens. And I'd like to, if I may, can I thank our president here?
Yeah.
Because he picked up on some information somewhere that ultraviolet light in the body would be helpful, and he's 100% correct. You can try to make fun of people. You can do anything you want, but I have a stack of research articles that show that he's 100% right. We're on the right track. We're going to get to the answer. We're going to resolve this.
And it's through your associations might be the reason he knows this information, right? So some of the people you're associated to that pass that
Could
forward.
be. We've been fairly quiet, but we're doing some research in other parts of the world, and I think soon we'll have some evidence that our president can use to support his case
Yeah.
against the naysayers, and a lot of them are experts in the medical community.
And we encourage people to go out and do their own research and find out about this information because it's out there. It's most commonly known as UBI, ultraviolet blood radiation, but the radiation works, so we call it ultraviolet blood therapy and polychromatic therapy, and it's real science, and I encourage people to go out and
You
look
can
it up.
go to the Library of Congress and look it up.
Yeah,
It's all over the
there.
place. It's been done in the United
all kinds
States
of-
prior to the current FDA administration, really promoting the drug companies and keeping device therapy in the background.
And there's, and at MatthewDeBow.com, I have several articles and videos about ultraviolet light and ultraviolet blood therapy systems. And if anybody wants more information, I've written a book, Light, Health Benefits and Medical Applications. Thanks for coming, Gene. I really appreciate all
Thank you,
your
Matt.
information.
Appreciate it, buddy.
And stay in touch.
God bless everybody and be well.